In this video, we're going to talk about viral morphology, which is the structure and especially the shape of different viruses. So first, let's talk about some of the basic structures that are found in some, if not all, viruses. So first of all, a single viral particle is known as a virion.
All viruses have a nucleic acid genome. Remember, a genome are just the genes that provide the information needed for the virus or organism to do what it does. And these genes help to code for the production of proteins. And one thing that's different about viruses, different than things like bacteria or eukaryotes, is that the genome might be DNA or it might be RNA. So that's something that's kind of different. The RNA genome is something that you don't see in bacteria, plants, animals, et cetera, et cetera. And on this picture here, this is an HIV virus here. You can see the RNA genome is this kind of swirly bit in here. Virions, or virus particles, also have a capsid, and the capsid is a bunch of proteins that surround that genome or that genetic material. So you can see the capsid here, and it's these proteins that are kind of forming a protective layer around the genome. So you could maybe call it like I don't want to call it a nuclear envelope because it's not that. It's very different structurally from what you would see in like a eukaryotic cell, but it has a similar function in that its goal is to kind of package and keep that genome safe. Now, some viruses have an envelope, but remember, this is not all viruses. The envelope is similar to a cell membrane, so it's usually made up of things like phospholipids, and it is the outermost layer. So you can see here, this is the viral envelope here, and it's this kind of additional outer protective coating that is kind of superficial to or more outside of the capsid. And then last but not least, some viruses, so remember not all, but some, have these things called glycoproteins. And these glycoproteins are these spikes that are sticking out here. And if you are familiar with the coronavirus, COVID-19, SARS-CoV-2, which we kind of all dealt with a pandemic just a couple years ago, you probably heard about spike proteins associated with the coronavirus, and that's these glycoproteins. And one thing to, if we kind of look at this word glyco and protein, so glycoproteins are just molecules that are made up of some carbohydrates. So glyco means carbohydrate, and then protein obviously has to do with proteins. So these are just molecules made up of proteins and carbohydrates bonded together. And we'll talk more about how these glycoproteins help a virus attach to its host a little bit later on. So let's take a moment and review what this term genome means. So if you took a kind of a different general biology class, typically like a first semester class as a prereq for this one, you probably learned a lot about these different terms. You probably talked about DNA and chromosomes and genes and the genome. And all of these terms kind of go together, but they are not the same thing. So let's talk a little bit more about what we mean when we say that viruses have this genome to them. So first of all, the genome is kind of the all-encompassing term of these other terms. So a genome is just all the genetic information in an individual organism. Or in this case, remember, viruses are not living organisms, but they do still have a genome. So for example, in humans, the genome consists of 46 chromosomes. And so essentially, all of your cells, with some exceptions to that rule, but most of your cells have an entire copy of your genome. It's got the 46 chromosomes made up of DNA that house the genes that you need in order to exist, basically. So the viruses also have a genome, and this genome does consist of genes. And these are genes that the virus needs for survival. And these genes might code for proteins. So remember, a gene is basically just like a recipe that tells the cell how to make proteins. And in viruses, these proteins might be associated with the capsid. They might be associated with those spikes sticking out of the virus. It might make enzymes, so et cetera, et cetera. Now, viruses have a much smaller genome than humans. So in humans, we have about 3 billion base pairs. Remember, a base pair is looking at things like adenine pairing up with thymine and guanine pairing up with cytosine. And so we have 3 billion of these base pairs that tell our cells what to do and how to survive and keep us alive. Now, viruses, we covered in a previous video, are very, very small. And so their genome is typically less than 300,000 base pairs, so significantly smaller than the human genome.
Another interesting thing about viral genomes is that not only can they be made up of DNA or RNA, they can also be single-stranded or double-stranded. And this refers to single-stranded RNA or DNA and double-stranded RNA or DNA. So in the past, you've maybe heard that DNA is double-stranded and RNA is single-stranded, and that is true for a lot of other organisms, but in viruses, the genome, we can actually have single-stranded DNA, double-stranded DNA,
Single-stranded RNA or double-stranded RNA. The genomes can also be circular, which is kind of like what we see in prokaryotes like bacteria, or they can be linear, which is something that we would see in eukaryotes. They can also be continuous, meaning that the genome exists as one piece of genetic material or nucleic acid, or they can be segmented. So there might be multiple pieces of genetic material that make up the genome. Now, these differences in the genomes can actually affect how a virus does what it does. So, different types of viruses are able to infect different things, and they're able to kind of replicate within their host in different ways, depending on these differences. Viruses can also come in different shapes and sizes. And so, we're going to talk about these 4 basic shapes of viruses that we can have. There are enveloped, filamentous, icosahedral, and head and tail. And we'll talk about each one of these in more detail on future slides. So first, let's talk about enveloped viruses. So remember that we talked about the kind of the basic structure of a virion on an earlier slide. And we talked about how there is genetic material, RNA or DNA, that is surrounded by a protein capsid. And then outside that capsid, there might be another structure called the membrane, or called the envelope, which is a membrane. So remember, we've got our genetic material, which is going to be our RNA or DNA. We have our capsid, which is made up of proteins. And then our envelope is going to be made up of lipids primarily. And you can see in this picture here, you can kind of see that envelope or that membrane surrounding this virus. Now, a virus that is not enveloped can either be called non-enveloped or it is referred to as a naked virus. Filamentous viruses are these kind of long, skinny rods. So this is a filament or one viral virion particle. This is that tobacco mosaic virus that we have talked about previously. And so each one of these is a virion that is filamentous in shape. Icosahedral refers to a round or geometric shape. So you can kind of see the core shape here is this kind of round or spherical or kind of geometric shape to it. And then head and tail viruses, or the head and tail viral shape, is actually the filamentous type shape bonded to an icosahedral shape. So you can see here we've got the kind of the filamentous area here where the tail is. And then up here we have the icosahedral. And these are primarily made up of bacteriophages, which are these viruses that infect bacteria. So we've seen this kind of black and white microscopic picture before, but remember that this is a prokaryote here, so probably a bacterium. This is the cell membrane of that bacterium. And then this right here is the bacteriophage. And to me, they look like alien spiders. As you've seen, viruses have a wide diversity in their morphology. So what they look like, what their genetic material is made up, what they infect. So it's a very, very diverse group of non-organisms, if you will. So there are more than one ways to classify viruses, and it just kind of depends on who is doing the classification. So they can be classified based on the type of nucleic that makes up their genome. So once again, we can have the single-stranded DNA, the double-stranded DNA, single-stranded RNA, or double-stranded RNA. They could be classified based on their capsid structure. They can be classified as to whether they are enveloped or non-enveloped, which remember we also called naked. Or they can be classified based on their genome structure. So is their genetic material linear, circular, continuous, segmented, etc., etc.? One commonly used viral classification is called the Baltimore classification. And you can see here that there are 7 different classes listed. And these classes are based on the type of genome. So whether it's a double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA. And also its mode of replication. And we're going to talk more about how viruses replicate in a different video. But ultimately, all viruses have to replicate their genome in order to make new viruses. And in doing so, they usually have to not only make more genetic material, but also make proteins so that they can make more capsids or spike proteins. And so you don't have to memorize this chart here. You just need to know that these 2 factors go into this classification. So the type of genome and then how it replicates.